The general structure of a biological membrane is a double layer of phospholipids. Other lipids and diverse proteins are embedded in this membrane or attached to its surface. But we do find that this is a semi-fluid structure. Okay, so if you were to take two different cells with labeled proteins and merge them together, over time, those proteins would flow and shift and move. So this is not a sort of rigid structure. It's constantly flowing and moving. And the more unsaturated the phospholipids, the more fluid it'll be, because that's going to prevent the tails being bent by the double bonds will prevent packing. Okay, in an animal cell, we typically use cholesterol to make it more rigid when we need to. Plant cells will typically alter the composition of saturated and unsaturated as they need to. But the overall fluid mosaic structure is what we see here. Most of the lipids and some of the proteins drift laterally in the plane, but rarely sort of flip-flop across. It can happen, but it's really, really rare. The active enzymes and the appropriate permeability, they must maintain that fluidity. But you have the proteins that are going to allow communication and transport. You have the microfilaments that are going to provide structural support. You're going to have carbohydrates on the outside that will connect to the extracellular matrix. That will be consisting of proteins like collagen, the glycoproteins connecting between the sugars and the proteins of the mosaic, and then the glycolipids connecting between the lipids and the extracellular. So we see the overall structure here. The fluid mosaic, though, it is constantly shifting and moving and flowing. So this is going to be a variable structure that changes over time. How do we actually make the membrane? Well, we have the endoplasmic reticulum, which makes lipids and proteins. Those will be then transported as a vesicle to the Golgi, where it will then be modified, and then that vesicle will flow through the Golgi, where it will then merge with whatever membrane is being produced. The inside of the vesicle, which you can see here, will become the outside. So the vesicle will fuse, it will open up, and so all of those surface proteins came from the inside of the vesicle, already embedded and pre-structured. We'll typically also transport things for export at the same time for a sort of double benefit. Why not? Okay, so that structure is going to be where it comes. When the vesicle fuses, it's going to release that structure, and the inner cytoplasmic layer of the vesicle will become the outer layer of the cell. What do the proteins actually do? They do lots of things. Like all proteins do lots of things. They're going to allow transport. Enzymes are proteins, signal receptors, recognition to the extracellular matrix, junctions and joinings. Those proteins are going to allow a great deal of structural and functional connection. The integral proteins that go all the way through are going to be transmembrane. They're going to be made of both polar and nonpolar structures, and they're going to be more involved with things like transport. Whereas the peripheral proteins that are on the outside, they're not embedded. They're typically going to be more polar in nature, and they're going to be more for communication and reception. There is selective permeability to the phospholipid bilayer, to this fluid membrane. So that selective permeability is going to allow some substances to cross more easily than others. It's a thin barrier, it's only about 8 nanometers thick, but specific proteins facilitate the passive transport of things like water and selected solutes, that kind of thing. But if it can get through the membrane on its own, it's typically going to be small, uncharged molecules. Larger molecules or charged molecules are going to need help to get through. So that permeability, simple diffusion straight through, needs no help. If it can diffuse, but it needs a protein to help it diffuse, we call that facilitated diffusion. If it uses a protein to diffuse, but it goes against the concentration gradient and requires energy, we call that active transport, which we'll get into more detail as we go later on. Passive transport is the more common type within this sort of base structure because it's energetically efficient. Things are going to diffuse randomly until you reach a dynamic equilibrium. The material will flow until there is an equal back and forth balance. Some things will be able to pass through, some things will not. So the things that can pass through will have to move and balance the things that cannot pass through to actually get to an equal concentration on either side of your membrane. So if you were to say, look at this example here, these large structures cannot get through the membrane, whereas the small ones can. There's more large ones on the X side, there's more small ones on the Y side, so I'm going to have to move more of the small ones to this side to make this an equal concentration. I'm not using any energy, so this is diffusion and osmosis, this is passive transport, okay, but we're going to have that relationship. So if we were to say that there was, I don't know, so if this is A and this is B, on X and Y, we have A and B, okay? So let's say that I have, I don't know, 1 molar concentration of A on this side, and I have 0.5 molar concentration on this side, and this one, there's a lot of them, so let's say, I don't know, a 2 molar concentration, and we'll do 0.5 on this side. Okay, diffusion happens first. You diffuse everything that you can diffuse. So diffusion, well, A cannot diffuse. A can't get through, but B can. So B is going to go from Y to X because there's more on this side. It will diffuse. Well, I have 2 and I've got 0.5, so that means that I've got 1.5. So that means after diffusion, I'm going to have 1.75 molar on each side of B, because diffusion balances whatever can balance. Okay, osmosis looks at the total. So osmosis is going to say, okay, well, if I look at osmosis, that's 2.75, right? And that's 0.5 and 1.75, so I've got 2.25. So if the B molecule was water, what's going to actually happen, I'm sorry, if A and B are suspended in water, what's going to actually happen is that, well, I'm going to need more water to go to the X side so that the concentration, the molarity, the moles per liter will then be what I need them to be. So the water is going to flow through osmosis from Y to X until the molarity is equal. These numbers won't change, the 1, the 1.75, the 0.5, and the 1.75, but the liters will change, allowing for an equal concentration gradient. So whenever you do these problems, do diffusion first, then do osmosis. But osmosis depends upon the sum. Diffusion is individual for every subcomponent. Okay, so that's going to move things back and forth, and that is going to alter the water balance and the tonicity. So solute concentration and membrane permeability is going to alter the tonicity, which is the ability of a surrounding solution to cause a cell to gain or lose water. Tonicity is a comparison, so you have to have two solutions. If the concentration is the same both inside and outside, that is an isotonic solution. If there is more material in the fluid outside of the cell than inside of the cell, then it is hypertonic. If there is less, then it is hypotonic. So water is going to flow through osmosis from wherever there is more water to wherever there is less water. So a hypotonic solution has less dissolved material, but it has more water. It has less solute, but it has more solvent. So in a hypotonic solution, water will flow into the cell, which we can see in the examples on the left. Plant cells prefer a hypotonic solution because the water is then going to push against the cell wall, providing strength and support. We call that turgor pressure, so the cell is called turgid. Animal cells don't have a cell wall, so our cells are going to swell, and if the swelling is severe, the cell can even break or lyse. Isotonic solution is the preferred for animal cells, whereas plant cells, they can function, but they don't like it because there is no pressure from the cell wall. The cell is flaccid. Think like a limp stick of celery. It's not good celery, but it's still celery. Okay, in a hypertonic solution, nothing likes that. So think really salty water, like the Dead Sea, for example, is extremely hypertonic. That is going to cause the cells to shrivel up because there's so much more solute outside, all of the water flows out. In an animal cell, it's going to shrivel or crenate, and that can be deadly. But for plant cells, this is incredibly bad because the cell wall is going to stay where it is because it's semi-rigid structure, but the plasma membrane tears away and all of the cytoplasm comes leaking out. Now there are ways to fix this, like an animal cell, for example, we can see the contractile vacuole is a special organelle that's in protists. These protists live in hypotonic environments, but as the water flows in, they have little muscle fibers around a vacuole. That vacuole fills with water and they shoot it out whenever they have too much. Plant cells prefer water to be moving in and out, so this is a good, but when they plasmolyze, the inside tears away and that cell is going to die, which is what's happening on the right. Active transport is a little different. Active transport goes against the concentration gradient and will require some form of energy. The most common example of an active transport situation is something like a proton pump. You're going to pump protons through proteins against the concentration gradient, so from low to high concentration. You might have other examples of pumps like the sodium-potassium pump. We use sodium and potassium powered by ATP to exchange back and forth, and that's absolutely essential for things like nervous system transmission. If you're directly using ATP to move materials against the concentration gradient, we call that primary active transport. If you are moving charged particles across a membrane, you will build up another source of energy, this electrochemical gradient, which you can see on the picture on the right. Positive charges build up wherever the protons are moved. You can use the difference between the positive and the negative charge to have things flow back into the cell. Like this example here is a sucrose proton co-transporter. So as the proton is flowing back in to balance the charge, I can also bring sucrose in for free without spending ATP. That's secondary active transport. So if you're using ATP directly, primary. If you're using electrochemical gradient, secondary. But active transport uses energy and flows against the concentration gradient. If I'm trying to transport large material, large components, food, lots of things, or really specific things, we're going to use vesicles for that. If the vesicle is going to grab things on the outside and move it in, we call that endocytosis. If the vesicle is going to fuse and then have things release, that is exocytosis. Now, exocytosis really only has the one word, but endocytosis, there are 3 different subtypes, and I'm sorry, this course has a lot of vocabulary. Endocytosis, the 3 major types: phagocytosis, which translates to cellular eating, penocytosis, which translates to cellular drinking, and then receptor-mediated, which is that controlled structure using protein receptors. Phagocytosis is incredibly rare. Very few cells are able to do this because things with a cell wall are typically not capable of this amount of flexibility. So phagocytosis, humans, white blood cells, for example, are phagocytes, that kind of thing. Penocytosis, almost all cells are capable of doing. It's small little vesicles that are going to allow that flow of material and suspended solutions, typically small solutes and dissolved solutes. Whereas receptor-mediated could be, it's basically penocytosis, but it's controlled. You have a lock and key mechanism. So only specific things are going to be brought in that way. And there may be sort of contaminants or cross issues like we can see here, but there will be some sort of trigger that will bind for that structure to occur. Okay, endocytosis and exocytosis occur continually in most eukaryotic cells, but the amount of plasma membrane remains fairly constant. Basically, one process will offset another, but if you need to move large things or you need to move things incredibly quickly, vesicular transport is the way to do it. This does cost energy. It will require motor proteins to move things around, so it's still technically active transport, but it uses a vesicle, so vesicular transport, aka bulk transport, is what this one is. Hope all that made sense.